Bueno, primero que nada, bienvenidos. Muchas gracias por conectarse. Pues la verdad sí me da mucho gusto tener gente de otros países, de otros continentes, de otras entidades de la UNAM. Pues bienvenidos a todos. Y para mí es un placer de presentar Mauro Degli Esposti. Él mismo se describe, ahora es una cita traducida, como un bioquímico que está vagabundeando por el mundo y dice que se requiere un libro para describir todas sus historias y anécdotas. Voy a, como sus estaciones en la vida, las voy a como reducir a unas pocas palabras. Él trabajó como profesor en Bolonia, en Italia. Después hizo 2 estancias postdoctorales en Rice University en Estados Unidos. Fue Senior Research Fellow at Monash University en Melbourne, Australia. Hizo otro postdoc en el Reino Unido, no especificó acá. Después fue Research Fellow and Lecturer en Manchester University en el Reino Unido. Después regresó a Italia como Senior Scientist en el Italian Institute of Technology. En 2016 vino por primera vez al CCG para una estancia sabática y yo creo que recordarme que te fuiste y regresaste, algo así, y ha trabajado en 5 países en 3 continentes y ha construido, apoyado en la construcción se ha unido a 15 diferentes laboratorios. Ha publicado más que 120 publicaciones y tiene más que 9,000 citas. Hoy nos va a presentar el seminario llamado Evolution of Cytochrome, Oxidase in Eukaryotes. Y pues Mauro, it's a pleasure. And pues please go ahead. Wait, mate, maybe before you start. So for the question, para las preguntas, les pido que si tienen una pregunta que lo escriban en el chat o que pongan, tengo una pregunta y yo les voy a dar la palabra al final y voy a leer las preguntas a Mauro o poner como para que tengamos un poco de orden. La gente que nos sigue en YouTube también puede poner sus preguntas en el chat de YouTube y lo vamos a copiar aquí, lo vamos a leer y lo vamos a contestar. Bueno, pues muchas gracias y perdón, now it's your turn, Mauro. Please go ahead.
Muchas gracias. Estoy esperando. Ok. I have to start in Spanish. Bienvenidos todos, colegas del mundo, no solamente de México. È un placer hacer questo seminario, però questo è il termine della mia parte in spagnolo. Seminario, all the slides are in English and the seminar will be in English. Now, the title is Evolution of Cytochrome Oxidase, which has been the subject of my research in the last few years. And also eukaryotes, because, well, whenever you study, this is the respiratory enzymes of mitochondria, and if you study mitochondria and their evolution, obviously, you will have to study and understand the evolution of eukaryotes. These are beautiful pictures that my colleague from Chile, Raquel Catrini, sent me recently of a beautiful place, which are favored to collect the various bacteria we're talking about. And here we have on the right, my reconstruction, what could be the oldest cytochrome oxidase, also called complex 4 of eukaryotes, of C. merulae, a small, very interesting red algae. Now, cytochrome oxidase, as Diego is part of the audience, and I'm very happy for that, he made already a wonderful presentation of the cytochrome oxidase, que is the last enzyme of the respiratory chain, the one actually consume oxygen, and tradition has been called complex 4 from the work originally done in before mitochondria. 1, 2, 3, 4. And this in the bottom is just an appetizer, what is going to be the complex evolutionary panorama. It's like a fan of the current understanding of the evolution of eukaryotes. Somewhere here, Lika, last eukaryotic common ancestors, and then you have two major branches with plastids, then become photosynthetic, more or less spread on the left, and the various other organisms which are not photosynthetic, including us, which are part of what is now called amorphia. The evolution of cytochrome oxidase, as I said, is my most important project of the last few years, and I already got some outputs. This is a major paper in bioarchives with multiple colleagues and approaches. The phylogenetic part of this paper hasn't been published yet because it's under revision, but in the meantime, they asked me to write a review on the topic, which is here. And more recently, there is an associated topic by the group of Rachel Quattrini in Chile, in which I am also honored to be a co-worker. It just been formally accepted in ISME journal yesterday. And the very first paper, I made this artistic view of the reconstruction of the time and where. We know certainly where, we know how. It's difficult to know exactly which were the bacterium that originated cytochrome oxidase, hence the respiration as we know it. But it's a good hint is that it happened in a place like Atacama Desert, but was full of water. Lacustrine environment in which the original cyanobacteria, oxygen evolving bacteria were freshwater. They had blooms there, and there was a lot of available rocks rich in sulfur and iron, pyrites. And then somehow these bacteria, originators, and most likely related to what the current Aciditobacillus is, invented a way of exploiting this excess of oxygen to oxidize iron and produce their lifestyle, the lithotrophic lifestyle. A consequence of this lithotrophic kind of physiology is what is called bioleaching. We can reproduce this in culture. We have an accumulation of trace metal like, in particular cobalt, in particular of interest for mine companies because it can be exploited at a very large level. Here you see here, this is what is called a heap full of mine tailing, not very rich in copper, but enough copper that the bacteria, the Aciditobacillus and others are grown on top of this, and they leach in an acidic liquid lots of copper, which again collected in this place, and then it is used for recycling and increasing the production of copper from mine. To give an idea of the size of this incubator, if you want to know, this open air incubator, this little thing here, it corresponds to the size of these huge tracts of mine. 25%, I understand, of the current production of copper in Chile is made by bioleaching. So there is an important application of this whole stuff. And I realized only recently that actually is a place, at least one place in the Andes, Argentina, not far away, I think, from the Atacama Desert, which is resembling what the artistic view of the old 2.3 billion years old emerged land environment, which we think from the evolution of cytochrome oxidase started. Earth was very different then. It was actually a combination of different current bits and pieces of different continents merged in one was called Canorland. But this is just curiosity, and is also a matter of speculation among geologists. But we know very well, because of the trace elements and very detailed geochemistry, is a geochemistry, the development of oxygen on Earth. It started approximately 2.5 billion years ago, producing by the oxygen evolving cyanobacteria produce what is called the great oxygenation event. And then the oxygen level, más o menos, approximately 1% of the level of nowadays, so talking about 0.2% oxygen, stayed like that, and is basically at the equivalent level of the Pasteur point in which yeast starts becoming all anaerobic. It stays for over 1 billion years, and then somehow, sometime between 600 and 800 million years, most likely because of the development of much more sophisticated photosynthetic eukaryotes producing oxygen, we are at the level of now. So animals, the separation of animals from the other branches of eukaryotic tree occurred among this time of period. Before that, we had another very important, two important, the mitochondrial endosymbiosis created what LICA, last eukaryotic common ancestor, are the only mitochondria. And then this predatory phagotrophic kind of organism is supposed to have eaten some cyanobacteria that were floating around, not much later than that, a few hundred million years, and then establishing the red algae, first of all, Rhodophyta, and several other algae. And in parallel, those didn't get this plastid, they become what we are as a species nowadays. So, having almost completed the project of the evolution of cytochrome oxidase at its origin, now I'm going back to the original project that brought me here in Mexico, back to mitochondria and their evolution from alpha protobacteria, already mentioned by Diego. The evolving hypothesis actually now there's our marine alpha metagenome acquired genomes close to Rhodospiralaceae could be the living ancestors or close to the living ancestor of mitochondria. And the strategy to analyze this is using all the knowledge acquired on cytochrome oxidase, analyzing in detail, and to look at complex 4, cytochrome oxidase, and looking for the aerobic ancestry of mitochondria. But it's a bit, well, as you say, strange that I come to Mexico to find the living ancestor of mitochondria, and I certainly found the living ancestor of plastids. The living ancestor of plastid, discovered by David Moreira a few years ago in this beautiful lake called Alchichica near Puebla, is called Gliomargarita, a deep branching cyanobacterium full of, as you see here, full of enclosures of various calcified elements. It's producing living stromatolites in this place. And when you look at the phylogeny, Gliomargarita is just a sister of all the plastids of the Archaeoplastidia, the three major lineage of algae which are considered to have primary plastids, which means they are close to the first endosymbiotic event that produces these photosynthetic organisms. Here is just a more sophisticated pictures to show you the impact of Gliomargarita or related Gliomargarita acquisition in the eukaryotic lineage with the Archaeoplastidia Rhodophyta, Viridiplanta, which includes all plants, by the way, and the minor Glaucophyta. And what happens afterwards, and that's been happening not long ago, in terms no more than 500 million years ago, that some organisms have started eating some red algae and then become secondary symbiosis. All these organisms on the left of the fan of the pictures have got, and then some actually have lost, a red algae and then red plastids associated with it. On the right, only we have Euglena, who has actually got the green type of chloroplast for some green algae. And here on the right is my simplification of this very complex picture of today's eukaryotes, and the major lineage are Archaeplastidia, these guys here with primary plastids. Then on the left, SAR, most of these organ, not all these organisms, they're also the Cryptophyta, are derived from secondary endosymbiosis, so they are not really interesting for me studying the origin of mitochondria and eukaryotes. And on the right here, we have the non-photosynthetic groups from Metamonada to Metazoa and Haz. Well, we have very good evidence that the oldest eukaryotes we can study so far are red algae, because of beautiful fossils uncovered a few years ago in India, which are 1.6 billion years, 1,600 million, and they are related to multicellular type, because Rhodophyta, red algae, can be also multicellular. So we can say that in this big branch Archaeplastidia, the very first leaf is made by red algae. And among the red algae, they are also unicellular, acidophilic. Mostly they also are thermophilic. They live in hydrothermal vents or situations in which a lot of sulfur, a lot of hot and acidic, which gets some names. One in particular, Galdieria, from an Italian botanist of the 19th century. And it comes in two different flavors. The genus Galdieria, which is the biggest, is also can be grown heterotrophically, is very adaptable. It's one of the most adaptable eukaryote you can think of. And that contains many mitochondria, see all the little arrows. It's about 10 micrometer wide, and they're much more common, cyanidium type, much smaller cells with a single mitochondrion and a single huge plastid. And these organisms are considered the earliest branching, consequently the oldest of the red algae. And they live in particular in places like this, and Naples, around Naples, there is the largest caldera of Europe called Campiflegrei. You have a picture here from my colleague from Naples, Claudio Ciniglia, who kindly show you what kind of a hellish environment is still there. And the green formation, you can see this, the green formations in the rocks are made by living cultures of Calderia sulfuraria. Of course, everybody knows that this area had another volcano called Vesuvius, and we have a very good record of what happened the last time it really got an eruption. But this, as I showed you before, is ongoing all the time. So these are endolithic algae. And the other thing, despite they are part of the red algae phylum, they are not red, they are green. And they are green because they lack the phycoerythrin, the typical red pigments of red algae. And this is another one. This is present also in the same environment, not so common, has been named by some Italian colleagues who I don't understand why they put such a Cyanidius Kislon merolae, very complicated name for a very simple cell. And from now on, I will call it C-merole because I will get difficult. And it's a fantastic little organism with a single mitochondrion, a single nucleus, a little bit of a Golgi, and a huge plastid, and divides by, fission asexually in this way. In fact, it has become a very good model for people like to study how organelles biogenesis. Not only small, it also has very few genes. About its genome, published in 2004, contains 1,000 unknown protein and 4,000 known proteins. So it has the same size, more or less, as the Rhizobia that are studied as CCG. And there are a number of reasons for that. But the interesting thing is, among these genes, I found, because actually resequencing in silico the whole genomes, because it's incomplete, in particular for cytochrome oxidase subunits. And I found a number of them, as you will see later. And then came across this paper of 2020, in which they sequenced 5 of these early branching acidophilic red algae, and came across this deposit in GenBank, Cyanodiophyceae, actually Cyanidiaceae, species MXAZ01. And you look at the record in NCBI, has been sequenced and deposited by Luis Servin, and I think this must be Esperanza Martinez Romero, 2014, just before I arrived here, and never published. And I hope we should soon publish this genome, probably as Cyanidinium americanus or something similar. Well, this was just found recently. This particular Mexican Cyanidinium comes from Los Azufres, which is a place with hydrothermal volcanic area in Michoacan. And believe it or not, it was already explored many years ago by the same group, including Professor Merola, the one who gives the name to C. Merola, and described organisms very similar to the one living around Naples. And this is a phylogeny of extant Rhodophyta, red algae, made up by the mitochondrial DNA, by concatenation of mitochondrial DNA coded proteins. And you see Cyanidiaceae occupy the basal position. Then you have the Bungiophyceae, another. They're all multicellular. You must have at least eaten some of them, because if you eat sushi, you eat some red algae that wrap it up. And these are related to the fossil I showed you before. But the great majority of species are all from the sea. They are Floridiophyceae, and there are over 7,000 of those. Not as interesting as the Cyanidiaceae. And now you see my own first tree of this presentation, which I've done the phylogeny of extra Rhodophyta using a subunit, nuclear encoded subunit of complex 4, which is called COX-5b. This is a very interesting little protein, not very long, about 150 amino acids, for 3 major reasons. First, it's soluble and sits on the matrix side, internal side of the complex, and interacts predominantly with the C-terminus of COX-1, of which we have an expert here in the audience. And this C-terminus just inserts itself right in the back of this beta sheet structure, which actually binds a metal not present here, it's called zinc. So it's got very unusual feature. It sits there, binds COX-1, and binds in a special way. And despite its relatively short length, it has a relatively good phylogenetic signal, because this type of tree is very similar to the one you obtain with multiple concatenated mitochondrial protein. What is new here, and my little discovery, is that there are actually alpha proteobacterial ancestors of this type of protein, never considered before. And so the common knowledge that the nuclear encoded subunits of complex 4 have no bacterial homologs is going to be different now, because they actually found these alpha proteobacterial ancestors. And with this and other proteins I found in genomes and C. Merole, I'm able to reconstruct here what it could be the 3-dimensional structure of complex 4 of C. Merole, and consequently, most likely, the oldest possible, we can think of, possible eukaryotic respiratory complex 4. It contains 8 subunits in all, which is only 2 less than the very recently published structure of complex 4 from plants, Vigna radiata, mung bean. which is similar in many respects, but also has got distinctive feature from those we knew before from mammals. And this is another view of the same structure, 180-degree rotated, COX-5b here, and to show you in comparison on the right with a high definition crystal structure of Paracoccus Genitrificans, a bacterial, alpha proteobacteria COX, which also has, apart from the 1, 2, and 3 major catalytic subunits, which also encode in mitochondrial DNA in eukaryotes, a small short transmembrane protein called COX-4, which is in position and topology very similar to either COX-7C or others which are present in mammalian and eukaryotic complex 4. So you can think these are kind of protective envelopes that be multiplied and then put together around the membrane part in particular, and of course the matrix and the other side, to make a kind of wrapping, packaging of the complex in eukaryotes. Well, this is plants, so talking Virdi planta. What about the evolution of green algae and plants? Well, there are lots of people working in plants in our institution. Well, they are known to be divided, I would say, traditionally in 2 major groups: the Streptophyta, which include mosses and vascular plants, and most plants we can think of, and the Chlorophyta, which are the green algae. More recently, they've introduced a possible daddy of all of this, a lineage containing Prasinoderma coloniale, which is a unicellular alga that lives and swims around in the ocean. And the genome of this Prasinoderma appears to be pretty old. But we have also for green algae very good and ancient fossils, and they are related to another multicellular algae, ulva. Again, in this tree, you will see how deep and how early Cyanidiaceae and red algae branch. And these are the beautiful fossils for Proterocladus antiquus, being named because of this close similarity to current Ulvaceae, green algae. And this is a lovely reconstruction of this algae when he was alive. And here, I think it's a much more sophisticated, but since we have taken time into consideration from the very first slide, this is an important and summary molecular clock analysis of all the organisms we have been talking about. So eukaryotes with plastids, starting from the GOE, created by relatives of Gliomargarita, they were already here, branching here. As you see, this branching of the cyanobacteria is much earlier than the group or the majority of current cyanobacteria, which branches after the endosymbiotic events that gave rise to the eukaryote, photosynthetic eukaryote. You see here the red, Cimerula right here at the bottom. Is actually in this paper has been underestimated the time of divergence of red algae. And it's actually more recent paper, they put it around 1.5, 1.6, at least 1.6 billion, because we have already evidence as fossil, 1.6 billion years old. First red algae, then the green algae, and then the plants. What about fungi? We had a discussion recently, we are both not experts of fungi. Fortunately, we have somebody in the audience who was an expert on fungi. And I always thought, because of my previous knowledge of the evolution and the pattern of eukaryotes, that fungi cannot be very old. But recently, they have been disputed, controversial, but published potential evidence they could be at least 800 million years old. But they cannot be as old as the algae I've shown you before, because they are part of the opistocones. I don't know many people in the audience know the word, it's difficult to pronounce, called opistocont, which means single flagellum. Has been known for a while that single flagellum is present also in some of the most earliest branching, like citrids, fungi. Most other fungi have lost it. And all these other organisms that up to metazoa, they have a form of their lifestyle with a single flagellum, like spermatozoa. And this opistocont lineage includes fungi, metazoa, and all these organisms. And then following the tradition, it was not my idea, and there was something else behind here. But I got inspiration from Diego Gonzalez. A similar slide, I used a recent actress as a representative of Homo sapiens, not Sophia Loren. And the opistocont legacy can also be seen looking at the three-dimensional structure available and the comparison between yeast, Saccharomyces cerevisiae, for which we have multiple structures, and recently acquired plants. Yeast has got 13 subunits, 3 mitochondrial, 10 nuclei. Plant has got 10 subunits. And those are missing. Look, they are all missing at the negative side of the membrane, except COX-5b, which is a really old member of the nuclein-encoded subunits of complex 4. And this is our complex 4. Humans apparently has got an extra subunit, but again, look at the lovely complex structure of COX-5b. Sits there, it's like a clamp, clamping together COX-1, COX-3, and a few others at the matrix sides on the membrane. You can also look at the evolution of Cytochrome oxidase by looking at the different composition and number of nuclein-encoded subunits. Not only that, and this is a special case for COX-5b, it changes its structure along the evolution with a major transition here between Philasterea, which are related, opistoconts related and sister to the animal lineage, and animals. In fact, animals COX-5b have a different structure. You can see here from this alignment. Ancestral, so this is a zinc finger protein with a single zinc metal, which is coordinated in the ancestral from bacteria to red algae to up to Capsapsora, to one cysteine, one histidine, and a couple of closely spaced cysteine. However, the animals, or the distance in time between these two lineages is less than 100 million years. Yet they lost, in this particular probe, they lost the zinc finger binding histidine ancestral, there's nothing there. And they acquired the new ligand in beef, animals, got this ligand with another cysteine. So strange the structure. And this coordination structure changes the relationship of this protein with the others at the matrix sites, and you can see their distinctive features. And so these slides essentially recapitulates or illustrates the new phase of my research in the evolution of mitochondria from bacteria, looking for molecular signature in the right COX-related protein. This is a COX, ancient COX subunit, for molecular signature like this, which you can define very well. And sometimes you see here, the histidine has been changed to an asparagine, which is normally a conservative change. My asparagine cannot really bind zinc. However, you see the pop-up of a cysteine right here in the same area, and maybe this compensating this lack of capacity on back. Anyway, this kind of precise molecular signature, which have reverberation at the structural level, you can actually define to see the difference between plant and mammalian and yeast just in the region of this particular subunit, and also have significance at the evolutionary level. So it's a combination of three different things, from sequence analysis, always been my expertise, to three-dimensional analysis and to evolutionary aspects. And this is also, room for a kind of mutation analysis never done before. As far as I understand, this particular subunit has not very much studied at the molecular level in terms of classical mutagenesis. And with this would have been my take-home message at the end of my seminar. However, something happened the last week or so. I became aware of two novel eukaryotic lineages associated with Archaeplastidia without plastids. They are predatory flagellates related to rhodophytes. So red algae are classically, they don't have flagella, they have plastids of certain kinds, they don't have vacuoles and things like that, so they have a specific well-defined morphology. Yet Rhodophyta and Picozoa are now considered to be related to red algae, and consequently they must be related to the earliest eukaryotic lineage which took on the plastids. Rodelphi has been published 2 years ago in this Nature paper, and this is a biflagellar predatory. In fact, you can only grow it in the presence of a prey here, and contains a plastid, but is an empty sac of a plastid. It hasn't got any of the light harvesting and the photosynthetic apparatus, no rubisco, yet it contains some indispensable enzyme for the biosynthesis of heme and the biosynthesis of iron sulfur clusters. This is very much the same situation you have in some anaerobic eukaryotes like Trichomonas or Giardia, which have only remnants of what used to be a proper aerobic mitochondrion without DNA. There is no plastidia DNA here either, but yet they retain as an organelles as a kind of biosynthetic machines for important aspects of the things, including iron sulfur clusters. And so this could be, well, obviously they had the normal plastid to the begin with, but what is amazing is they have got flagella, they have a predatory, which means the ancestor of all photosynthetic eukaryote, what you call it, mixotrophic, a bit of light, a bit of energy from plastids, and a bit of food whenever it is available. Even more dramatic is this tree from a very recent paper published in bioarchives last week. And we're able, a group of all eminent guys in the field, including Etema, Fabien Bourqui, and so forth, they were able to put together the genome of single cells, which are very small, of Picozoa. I don't know how many of you have ever heard. These are members of the picoplankton, i.e., the plankton you can filter out with a 2 micrometer filter. And this Picozoa, of which we only had a few scattered material before, genomic material, ends up without any plastid whatsoever, with a beautiful Golgi apparatus. I got the intestine. It's like a little man in 2 micrometer of size. They are found to be sister of Rhodophyta. They are part of the same lineage, and they are non-photosynthetic. So we have to change soon. This picture, which is already sophisticated, complicated, 2 years ago, the new tree of eukaryotes, we have to shift the Picozoa from this orphan branch to the branch of the red algae, Rhodophyta and Rodelphis, with primary plastids. And I can give you an idea of the importance of new genomes from the oceans. From the available mitochondrial DNA of only one, again, I have to make a complaint in general. Most of the recent interesting thing, Rhododelphis, Picozoa, Prasinoderma, genomes are not available in NCBI. They are kept in other accessible, but it's difficult. It's like very difficult actually to access, but you can, but they're not publicly available. If you do a blast, you will never find them. Anyway, we have a little bit of mitochondrial DNA with a single picozoa, which we also have images of mitochondria included. And when you make a phylogenetic tree, it's a preliminary work of ND5, which is the largest mitochondrial coded subunit of picozoa, ends up to be apparently the earliest branching. So picozoa in their little mini size could be among the oldest eukaryotic, and certainly this kind of weird mitochondria. Some people say they eat viruses. This kind of bacteria, this kind of organism live by eating viruses, which would be very useful nowadays, but anyway, we don't know for sure. Anyway, this kind of mitochondrion could be the oldest mitochondrion we can study. And for people live down there in Chile or Argentina, you know, it's a place where there's higher concentration of this picozoa is. It's the ocean between the tip, the Cape Horn, and Antarctic. 30% of the pico plankton is made up this old ancient eukaryotes. And let me thank the various colleagues here in Mexico, in particular Esperanza Martinez Romero, who is the person responsible to bring me here, and various colleagues around Mexico, present and future collaborators. Some of them are present at this. And thanking the various collaborators around the world. Some of them, I know that Raquel gave me this beautiful picture, and Anna Beltran is the other co-workers in some of the papers together. You see the smile of Rachel? I think if I can see her now, the smile is bigger. Her big paper in ISME journal has been published yesterday. This is Lars, no published, accepted. Lars Hedersten from Lund University, an old colleague and friend. Claudio Ciniglia is a recent acquisition in the collaborative network, and Anna Gelli is one of my oldest and most accomplished students. And with this, muchas gracias todos.
Well, thank you very much, Mauro. Muchas gracias. Pues vemos a ver si hay preguntas, if there are any questions. You can, well, como les dije en YouTube, o pueden escribirlo en el chat de YouTube o en inglés o en español. Aquí ya. Sergio escribe en el chat. Sergio, ¿tú quieres decirlo en tus palabras o quieres que lo lea?
Okay.
Bueno, no te escucho.
¿Me oye?
Bueno,
Okay.
Sí, te oyo.
Okay.
Sí.
Can you hear me, Mauro? Great talk. Excellent. Thanks. Well, this question is very general. The cytochrome oxidase proteins require other proteins for the assembly.
Yeah.
For example, that to deliver or insert the copper atoms. Another several proteins, no?
Yes.
This group of proteins, do you think this group of protein also had the same evolutionary process as COS? What do you think about?
It's a very good question. Yes, the biogenesis, I spared you the pictures of the biogenesis of cytochrome oxidase, of which we have an expert among the audience here, and the best studied in yeast. There are at least 29 COX that helped the assembly. Well, as far as I know, Cimerula has got COX-11, COX-10, and some of the others, which are SURF and which are SCO and others, which are essential for the biogenesis. And one of the reasons I'm studying, I haven't told you, there is no sign of COX-15, which is IMHA synthase. Well, either is a very different kind, or the mitochondria of red algae don't have Cytochrome A. It will be the first type, and that's why I'm pushing colleagues to grow a lot of algae in order to isolate mitochondria and characterize them.
Okay, thank you.
Tenemos Esperanza Martínez. ¿Tú quieres decir tu palabra, tu pregunta o la leo? Como tú digas.
Léela tú.
Okay, gracias. Esperanza asks, will you be able to obtain Picozoa?
Myself,
Yes.
I have great hopes in my colleagues down in Chile, if they have connection with marine biology. In principle, can be found in any oceans with the right filter, and that in principle we could, but you have to work it out. One of the interesting things, the way they isolated in this paper in bioarchives, isolated these small cells is by fluorescence sorting. They actually use mitotracker and lysotracker fluorescent dyes, which binds to these organelles bearing organisms. The kind of dye I have in the freezer, I'm going to apply for studying with colleagues in red algae. So potentially we could do it, but it's not going to be easy.
Okay. Esperanza also asks, well, another question is there evidence of recombination among chloroplast and mitochondrial genes?
Of what? Of a recombination?
Recombination among chloroplast and mitochondrial.
Not really, but there are combinations when the genes from the endosymbionts move to the nuclei. Some of biosynthetic pathway, which are both in the chloroplast and the mitochondria or the alpha protobacteria, they can be used either way. So you have photosynthetic organisms which use either the chloroplast pathway or the mitochondrial pathway. In some cases, like Rodelphis, it could be that you have mixed situations in which you use members of both pathway, of both organelles in practice to put together the biosynthesis of certain important elements. One case in particular is IMA, but genetic recombination, as far as I know, is rare or not well reported.
Okay, thank you. There's a question from Xochitl. So Xochitl, please.
Hi, great talk, Mauro.
Thank you.
So one question I have is, if in this Picozoa, do they have COX-5b?
Well, I don't have, I only have some mitochondrial DNA available. This paper is already in bioarchives, and they say they will deposit the genes whenever it gets accepted and we get the genomes, we have to look. I presumably, I hope so, but in my own, if not, then we know exactly when this particular, my hunch would be that it would be there, because if you got some alpha protobacterial ancestry, it should be in all original ancient eukaryotes.
Okay. I have another question. Can I do it?
Yes.
So in addition to this histidine that you observe that is present in some organisms, but in animals is absent, do you see any additional feature that calls your attention on this group of organisms regarding COX-5b?
Group of organisms, meaning?
Yeah, like it's in animals, for example. So in animals, you don't have this histidine.
Yeah, okay. No, in fact, they say, no, very good question. In fact, it's varying. For amoebozoa, they've got a shorter version of the C-terminus. Oh. And the most puzzling is actually green algae. The green algae have a completely different the folding is there, but the major system, in particular ligands, are missing. What happens is like they are substituted most likely by glutamic acid.
I see. Okay.
But it's always lineage dependent. What is nice about this particular protein is that whenever you see some molecular changes of significance, it's lineage dependent. So on that particular change, it's only in green algae, it's not in Prasinoderma, it's not. And the one of present in Amebozoa is only in Amebozoa. Then there is another, well, considered deep branching, but they're also very rapidly evolving. Naegleria, who is part of the Discoba, they have a different length. So they have the same ligands, but they have something, a space in the middle, which is only unique to that particular lineage. So molecular variants are there, but they are lineage dependent, which is just good for evolutionary terms, I think.
Right. Okay, thank you.
Well, there's a comment by Paola Bonfante. She wrote, "Dear Mauro, great talk. Thanks. I have to leave." Paola, so she's.
Well, thank you.
Esperanza has another question. She wrote, "Are Picozoa representative of pre-chloroplast organisms?"
So say it again. Sorry, I missed the beginning.
Are Picozoa representative of pre-chloroplast organisms?
Ah, that's a big question. Either they lost it or they never had it, and this is clearly put in this paper, and there are no answers at the moment. It looks like they never had it. If they never had it, and this work is confirmed by others, it means there was not a single endosymbiotic event with Gleo margarita. There were possibly multiple similar, otherwise you wouldn't have this kind of situation of a sister which has never seen plasmid. So plastids is a real game changer. It's something we have to look very carefully because some developments will come up, I think.
Okay, thank you. Are there other questions? Please write down if you want to ask something. Well, I have a question, not much to do with your talk, but taking a chance because you're a mitochondrial expert. I was always wondering these kinetoplasts, or how they're called. They're ones from some unicellular parasites. I think it's from
Kinetoplastidia, you mean?
Yes, they have these kind of
Trypanosoma.
Composite genomes where you have small. You have these kind of the large. I forgot the name now, but there's a kind of a genome where they do a lot of this gene editing. So
yeah, Trypanosoma.
Yes, for example. And
yeah.
I always thought they are kind of deep branching or they're early mitochondria, but they are not apparently. Or
well, they are part of a deep branching group of the Euglenozoa, flagellates, Euglenozoa, but they are so much adapted to parasitism. We lost most of the mitochondrial DNA, most of the mitochondrial genome, and they are so much derived that, you know, of course, they have some of you have to take into consideration in a big tree, but well, I don't think they are in the real lineage of the well considered the primordial Lica. No, it's just there are some features, of course, in common, but they are too derived. I mean, they say Jacobida, which are relatively also part of the same lineage, which have the most genome rich, mitochondrial genome, most protein rich. This should be, you know, the kind of living mitochondrial organism, but most likely they have maintained this large mitochondrial compendium of proteins because they are very slow evolving. So some on the same lineage are fast evolving, like Trypanosoma, others are slow evolving. So it's very difficult to find the right balance there.
Okay, so I'm not sure my.
Well, there is one thing, if I might add.
Yes.
There is a very important human parasite which derives from a secondary symbiosis with red algae, and is called the agent of malaria. The plasmodium is an ex-algae. It's got a little remnant of the algae, and there's a red algae that was eaten by his predecessor. So even organisms you think nothing to do with plastids, they turn up to be related to some secondary symbiosis with red algae. I see a lot of comments here, no? Okay. Yeah. Is everybody happy? I kept it short, too short, or maybe no. Sometimes less is more.
I do have an additional question.
Yes.
Have you looked to the evolution of other respiratory complexes in addition to cytochrome oxidase?
In red algae, you mean?
Well, in general, like the kind of study that you are doing for
Oh yes, of course.
Because
yes, I'm planning.
In our lab, we have observed that you are looking that the COX-1 C-terminal end is important, and it has this COX-5B subunit interacting, but we have also observed that the carboxy terminal end of cytochrome B is also very important for biogenesis and general function of complex 3. And then there is this COX QCR7 subunit, which is also very close to the cytochrome B. So I don't know if you have looked at something like
I haven't personally looked, but I collected the genes and the material because, of course, the next step, reconstruct the BC1 complex 3 of C. merola, reconstruct the complex 1, which is not in yeast, but complex 1 as well, which is all the enzymes have been working in the past. So I can see there's a long-term perspective here. COX-4 is a big thing at the moment following the evolution of the enzyme, but I will certainly apply. We can collaborate on that if you want, of course, in BC1 complex and complex 3 in the future, of course.
Right. Okay. Thanks.
Okay.
Sergio?
Christian has problems with the internet service.
Okay.
So for that reason,
You're taking on.
Yes, but well, I don't know. Is another question for Mauro?
There are some comments.
Comment. What?
Okay.
Okay. Thank you. Thank you so much, Mauro, for this splendid talk, and congrats for your results and your research. And well, thank you to all of you. For
yes, very international. Thank you very much to everybody. Bye-bye.
Nice